cleaning and today about cleaning and storing and processing a lot of the native seeds that we have here. This is one of my seed boxes, and so I keep most of my native seeds and everything. In this box, I have a few others that are in this mini fridge over here. Generally, I'll keep them inside something that is permeable to allow water, vapor, and air to get into the packages. I've learned that if you keep some of these seeds in things that are airtight, that it's a lot more easy for them to rot in some cases, especially with like fleshier kind of seeds or fleshy fruits or like things that you haven't cleaned entirely. Magnolias are a good example of that. But most of the things that I grow personally are generally dry, kind of like dehiscent fruits and everything, so their seeds are not terribly fleshy or anything like that. So I've got like Cledrastris, this is yellowwood, some allium species and a few others, lots of milkweeds and everything. So I'll keep them inside these paper bags to allow for airflow to get to them and everything, and I keep them around like 45-ish degrees or so, pretty much the warmest temperature on my mini fridge. I also really like using these mesh bags. These are, I think I got them at like Michael's or like Hobby Lobby or something like that. And these are organza bags. They're kind of like meant for like wedding parties or something like that. And these are really useful both for storing seed and if you want to put them on top of like the developing fruits of something that you aren't going to be there to go and catch the seeds. You can just go back later in the season and all the seeds will have dehisced inside the bag. So you can just have this little drawstring just kind of cinched around the top part of the stem there with the developing fruits or flowers or whatever inside the bag itself. And they'll just kind of, drop down in there. It is very important to label everything. So with these bags, I just have like little paper labels that are in there. This is Cyprus echinacus. This is one of the sedges that I have. If I don't have a paper label in here, the label will be just, outside on the bag. I have a labeling system so that I can keep track of what I got where. So I'll have like the year. So this is 2023 G10A, and so this tells me I collected it in 2023. The provenance is garden, and so if I collected this from a wild population, this would say W instead of G. The number that I accessioned it as for this year, so this is the 10th accession for 2023, and then the individual accession, which is A for this one. So if I were to do something differently, germination-wise or treatment-wise for this, I would make another accession with a B and then C and D and so on. Generally, I don't go any further than A unless I don't really know how to germinate something, and so I'll give them like multiple different kinds of treatments. For example, with this Cyprus echinacus here, I wasn't really sure how to germinate this, and there was only one sort of documentation thing online that I found that was really any help at all. And so I made two different accessions, or actually three different accessions of it last year to figure out the best way to germinate this. And it turns out that having it cold stratify for at least 30 days or so was the best way to do that. So when I collected this last year, this is the progeny, the seed from the progeny that I grew. So when I collected this last year, I had some of it just directly sown into a flat, and then I had some of it directly into stratification for 60 days, and then some of it I just left outside. And the stuff I left outside actually did the best out of all those treatments. And so it seems like 60 plus days of stratification had the best or the highest germination rate overall. So yeah, organization is a very, very key thing to do. And so this is all seed that I either haven't stratified right now or I haven't begun stratifying yet, or seed that just doesn't need to be stratified in the first place. Otherwise, I keep them inside like plastic bags. For example, this is a plastic bag full of pawpaw seeds. This is Asimina trilova. that I got at the Arboretum where I work from a very strange plant. It's probably one of the cultivars from Kentucky State University's breeding program, but we just don't have any kind of data on the provenance of that. And so you can see the seeds that are in there. And I just have them in ProMix here. So it's ProMix mixed with a little bit of perlite, just to allow for, a little bit more air spaces in between there. And so these bags are made of polypropylene, which is a semi-permeable plastic, to allow for some airflow to get in and out of there. I have had very little problem with mold, especially using Promix as a media. If I were to use something just like straight sphagnum moss, that's usually when I get a lot of mold. So I'll keep them either in these polypropylene bags, or I'll keep them in high-density polyethylene in these little deli cups or deli containers. So you just, you'll get pimento cheese or whatever in these sometimes at the grocery store. I had a bunch of these already because I was doing tissue culture in them. And the media that I'll use for smaller seeds instead of the Promix is just sand. This is just construction sand that I got at Lowe's. It's like $5 for 50 pounds or something. I wouldn't recommend doing this type of sand overall. I would go for like play sand or something cleaner than this. That's just equally as cheap as the construction sand, but it's a little bit easier to deal with, and especially with photosensitive species. Since the majority of that sand is a quartz derivative, it will actually allow some light to get into the sand, so that if you have your seeds just buried a little bit under the stratification media, they'll still get some light just because it's partially translucent. And I'll talk a little bit about storage of extremely small seeds as well. And so these aren't native seeds, but these are begonia seeds. I also grow lots of different begonia species. At one point, I had over 300, which was pretty insane, and I would not recommend having that many at any point. These are Begonia variabilis, or one of the forms of it. And so these are coin cases, like coin collector cases that I got from, I think, Walmart or Amazon or something, as much as I don't want to buy anything from Amazon. So generally, the way that I store things in here varies maybe in two ways. And so you can see here I have like a little sheet of paper. This is glassine paper. It's super duper thin. Some people call it India paper. It's less than half the thickness of like copy paper or something like that. And so it both allows for airflow, because these are not airtight or anything. And so you can also just like sort of see through it to see about how many seeds are in there. You can see them just kind of rolling around in there. And so I use this for very, very small things like penstemon seeds, or some hypericum, some of the St. John's worts and St. Andrew's wort, and also St. Peter's worts also. Those seeds are very, very small. They're dust-like. So this is a good way to store things that are very, very small, or just things that you have very few of. And so for the process of cleaning seeds, it varies dramatically by just by the species. And so you'll have things that have very fleshy fruits that require a lot more processing to get the pulp away from the seed itself. Whereas many of our native plants, especially here in Kentucky and most of the Southeast, have like sort of dry, dehiscent fruits. This one included. So this is Silene regia. This is something that I've already know how to grow and everything, and I cleaned some last year. This was collected maybe like 3 or 4 months ago. Very nice, big red flowers. It's the royal catchfly is the common name for it. And so these have, as you can kind of see, this little dry kind of woody capsule that has an opening on the end of it. And that opening is where all of these seeds sort of spill out. So I just cracked it open there, and you can see those little black dots. And those are some of the seeds of this species. Because I've had it in this little organza bag for so long, many of the seeds have already dehisced themselves. out of the little pods and everything. So I'm just kind of crushing it up and getting most of that out. And what I'll do later on, I won't show this part, but it's I will use strainers or sieves or anything that has like a wire mesh kind of thing to get the seeds away from the chaff. So it is a good idea generally to separate the chaff and the seed, because the chaff is usually a lot more organic material that can cause rot and fungal growth, etc. Especially if you sow your seeds in a wet medium pro mix, etc. So most of these have already kind of fallen out here. I kind of just show that, but these are mostly seeds. There's just a little bit of chaff that's kind of already up in there. So the reason I'm crushing up these pods is because there are still seeds that have not dehisced themselves inside there, which you might not be able to see right now. Those little black dots that are way down in there are still seeds, and they're probably viable. They may not be because they haven't dehisced themselves, but it doesn't hurt to just get them out and try it again. So I made a little bit of a mess here, but I'll take all these seeds and chaff and everything and then run them through a series of sieves of different sizes to separate everything and get them ready for sowing or for stratification. Now, when you're cleaning things like fleshy fruits, this Magnolia, for example, this is Magnolia macrophylla, it's important to be able to distinguish the seed from the rest of the fruits or the rest of whatever it's in. And so with magnolias, one of the things that I do with these is I let them ferment at room temperature in some water for a couple of days, which really helps to loosen the aril, the little fleshy bit around the seed, from the actual seed itself. And so I've got a few of these in here. I've got about a fruit's worth in here, maybe a dozen or so. And these have all been fermenting for a couple of days now. So I'm going to zoom in just a little bit, if it'll let me. Here we go. So you can see already that there's a little orange bit. Oh, it actually just came off. There's a little orange bit here, and this was the aril. That's sort of what attracts the birds and stuff to disperse the seeds themselves. And then right inside there is this little black or brown kind of shiny oblong thing, and that's the actual seed itself. And so I'm going to go ahead and do that with all of these. I won't show every single thing, but then we'll talk a little bit more about recalcitrant seeds, which these are recalcitrant seeds. Now, basically, that just means that they can't dry out. So this is true of magnolias, this is true of sassafras, this is true also, I made this mistake recently, true of also of spice bush. These are spice bush seeds, and they dried out, and these are no longer viable, unfortunately. So they have to maintain moisture throughout the entire stratification process, or else they just won't germinate. The embryo inside them will die, unfortunately. So now that I've got all these clean, you can see about how many I have. This is maybe a little over 20 or so. And so after I separate all of the arils from the actual seeds themselves, I'll give them a little wash with some water through a strainer just to get all the excess stuff off. And then we'll put them in some stratification media. So when stratifying seeds, it's a good idea to just go ahead and organize everything so that you don't lose track of it later. So I've written on this little bag that I will be stratifying these seeds in, the name of the species, the date it will go in to stratification, and the accession number. So this is just how I personally keep track of what I have where. I have a little database kind of thing on my phone that I keep track of things there. Today is October 15th, so 10/15/23. And I know that this species, Magnolia macrophylla, needs at least 120 days in cold stratification. And so I'll write out, so 120 days would be 4 months from now, and so that would be February 15th. Okay, so 2/15/23, and I'll go ahead and put that on my calendar. And I know that this needs 120 days of cold, moist stratification. Right. So when you're thinking about the media for your stratification, if the seeds are large, like these Magnolia seeds are, generally I'll use a sphagnum heavy mix, and so this is just ProMix here. I'll just pour a little bit of water in there. But if you're going to use, or if you're going to stratify smaller seeds, things that are dust-like or just really difficult to handle otherwise, it's probably a good idea to use something that's a finer particle size than ProMix. I'm fond of using sand, quartz sand, play sand, something that's just a little bit translucent. Builder's sand would work. I use that for a lot of my stuff, but again, I wouldn't recommend it. And you want to make sure the moisture level is appropriate. So by that I just mean not too dry, not too wet. And so I like to do a little squeeze test, and so if it holds its shape for a little while, that's a pretty good moisture content for most things. You can get away with using very, very wet media for some things like wetland plants, which would be inundated for part of the year anyway, so they would be adapted for such conditions. Some species do require dry, cold stratification, but that is pretty rare, and only usually things like grasses or some sedges do that as well. So I'll put at least half the volume of media per volume of seeds. So it's half and half seed and media. This is, I just put a little bit more in here because I know that these, when I germinate them, will take up a lot of space each, and so I'm just going to go ahead and do that. So after this is stratified, I'll just sow them in a flat altogether with the media and let them germinate on their own. So just seal this up. And I'll put it in the fridge and forget about it for about 4 months. So now we're outside here in my little greenhouse, and I wanted to talk about some of the things that you can do after you germinate whatever it is that you have stratified or whatever it is that you're trying to grow. What I do with most of my plants, except for some woody plants, is I'll put them into plugs first, and so these are plug flats. These are I think SureRoots 45 cell plugs. This is a Dicanthelium species. This is Dicanthelium clandestinum. They've been in here for a little bit, and they've got pretty good roots so far. So these are air pruning, so the roots don't always go down straight to the bottom there. This is a good way to allow your plants to get a good root system and get a good head start on life before you transfer them either into the ground, or if you want to put them in a larger pot, you can do that as well. There's lots of different kinds of plug trays. These are little square plugs of the same species, and they've been in here for a little bit longer, and these are not air pruning plug trays, and so the roots there will need to be broken up a little bit before you plant them or before you pot them up. This is switchgrass. This is Panicum virgatum, and these are much the same. The roots on this species are pretty intense, so you'll have to do the same thing here. With most species, especially most native species, it's generally a good idea to get a deeper plug tray than one of these short ones here. So these are just little, practically cubes, and these are nice big tall ones. These 45 cell trays are generally, each individual cell is going to be a little bit larger than the typical 50 cell trays. I like these a lot too. I actually have some things germinating in here. This is Cyananthus, the fringe tree, is in here, and they need a period of warm stratification before they actually germinate. So most natives a lot of grasses especially. These are Andropodon ternarius and Indian grass and a few other species over here. They'll need lots and lots of root space, especially early on as they're getting established and everything. These have only been in here for maybe a little over 2 weeks or so, but their root systems are pretty insane for only having 2 weeks in these things. So the larger amount of soil you can give your plants will help them later in life. Now this is also true of things that you grow from cuttings as well, and so these are Euonymus americanus, strawberry bush, or hearts of Boston, or there's a few different names for it. This is in the family Celastraceae, same family as the other Euonymus and Celastris and the Oriental bittersweet and stuff like that. But this is a nice native one, and I've rooted these from cuttings. And I tried some in plugs, and they did not take, and these ones that are in the quart pots did take and are doing quite well. They were all taken at the same time and given the same treatments, but just the amount of soil, or the amount of substrate given to these, I think helped them establish better, and they didn't dry out as fast, and it was maybe a little bit easier for them to take a root overall. And the same is true of this little clinopodium here. This is something I grew from seed and then took cuttings of, and also this red elderberry, which I've already taken, and that's only been in there for about a week or so.